Andrea, thank you very much for accepting being with us here. Fortunately, probably the audience is not as high as we expected, but you know, after Eastern time, some people still are on holidays. So if you don't mind, I will give a short introduction about your career because it's amazing. I have to say you can be one example for many people. So just a couple of things, okay? So Andrea studied in the molecular life science at the University of Erlangen-Nuremberg. And you defend the PhD in 2011 from University of Göttingen in Germany on the topic of practical approaches to macromolecular X-ray structural determination with a group of George Sheldrick, right? Then you moved to run this READ group in UK, in Cambridge, working on I guess continuous working on crystallography, but very soon you became your independent fellow, Marie Curie, and you moved a little bit to cryomicroscopy. Then as a senior researcher, you were working at Diamond and University of Oxford and developed the software Auspex that probably many people have already used. You returned to Germany to work as a group leader at the University of Würzburg. That's a difficult one. In 2019. Also, you already implemented the artificial intelligence as a tool in your work. And currently, you are the leader at the Institute of Nanostructure and Solid-State Physics in Hamburg.
Yes.
Hopefully, I'll see you in June. I will visit it there. With this,
will you come there for longer?
No, only 2 or 3 days for a lecture in the bio.
Write me an email so we can have coffee.
Sure. So now I introduce your lecture today is lesson from the Coronavirus Structural Task Force for COVID-19 and beyond. So please, Andrea, the floor is yours.
Thank you very much. And thanks for the exciting invitation. Sorry for the technical problems we had previously. Can you see my slides? I hope.
Perfectly.
Yeah. Yeah, so lessons from the Coronavirus Structural Task Force. So I'm going to start pretending that you don't know about this. And I'm going to start a little bit more fundamental than a coronavirus, although here it is. That's what we think or thought it looked like until very recently. And of course, it is made up of molecular structures, some of which are macromolecules. And it are these macromolecules that we're particularly interested in. So schematically, we can display them like this, which is surface representation. But of course, this is a very large molecule. Actually, it's 3 molecules. And if you zoom in, they're all made up of individual atoms, thousands and thousands of atoms. And their positions are very relevant to what this looks like, but also which functionality it has. But we cannot display these molecules as individual atoms because, as you can see, it's totally chaotic. So for the purposes of this talk, we're only going to talk about surface representations most of the time, or perhaps ribbon representations like this one. And yeah, here they are. So first, what my group usually does. Where do our structures come from? These structures of large molecules that are so important for understanding DNA, understanding how biomolecular physics work. So we need to do a biological preparation. Any biological crystallographers in the audience will be very familiar with that. Then we measure data either by NMR, cryo-EM, or crystallography, mass spec, all the methods that help us. But then we have not a direct 3-dimensional picture of the structure. In order to get that, we need a model with which we interpret the data. It doesn't matter which method. We have no method that directly gives us a 3-dimensional picture. So we need to interpret the measured data with a model. And only if we can have such a model, which very often is an atomic model, do we actually kind of get answers to biological questions, right? Without the atomic structure, we know very little. Also, on the model depend all downstream applications. So I don't know if you know that, but 99% of the protein data bank structures are used by people who are not like experimental structural biologists. So things like molecular dynamics, structure-based struct design, bioinformatics, all depend on these models, which are deposited to the PDB. But these models are only an interpretation of our measured data. So problems look like this. So just so you know, this is coenzyme A. I know it's not immediately intuitive why this would be coenzyme A, because clearly it's like mismodeled. And if there are any chemists in the audience, which I hope, you will know structures don't look like this. In particular, there's no bond here, and there's like phosphates are all over. And that's like a real deposited structure from the PDB. That's a ligand in a protein, and that has been published. So it would be an easy choice to say it's the fault of crystallographers who are doing a sloppy job. But the problems go deeper than that. They also can lead to rejections in scientific journals, which is like the worst, right? It ruins your reputation as a researcher. But why do these errors happen? It's because of the fit between structure and data. So in crystallography, we look at this as terms of the R value, which gives us the difference between the data we measured and the data we would expect given a model. So this discrepancy is measured in percent. And this is a very typical experimental map at 2.6 angstrom resolution, and it has 20.4% R value to the model. And here is a perfect predicted map at the same resolution. And that, of course, would have an R value of 0% because the data in the model would fit perfectly. But we cannot get that. So there's no experimental structure in the whole world that has an R value of 0. We get some very good R values for small molecules like sugars, small organic molecules, 5% in average. They can be as low as 1%. But that's not true for the structures of larger molecules like proteins and RNA or macromolecules. Here, the average is 24% in the PDB. So 20 is a very nice estimate, actually. So why is the fit between model and data worse for large molecules than for small molecules? Have we missed some fundamental property of molecules that we should describe in our models? Are we processing our data right? Do we make measurement errors in protein crystallography that we are not making in small molecule crystallography? Are we optimizing the model mathematically well to the measured data? What's the role of the disordered solvent that makes up 50% of protein crystals? These are the questions that my group is usually dealing with. It's the question of, we know 200,000 macromolecular structures. How can we improve all of them and understand more about the nature of these crystals or these molecules? Because right now, the structures don't really fit the data. And that is where also errors come from. Because with a fit that bad, errors are really, really likely to happen. It's a complex problem. Fit between data and model are not perfect. Complete automation of structure solution is not yet possible. Models are mostly built manually. Expertise in many different aspects is needed. One of them is software, which is what we are working on. So I want you to keep that in mind for the rest of the talk. Errors in structures are still normal. And there is not a PDB entry that you can show to me where I can't find an error. We're doing our best and we're really doing excellent work, but the problem is very complex. So SARS-CoV-2 is made up of molecules and has this life cycle, which I'm going to show a little bit in more detail later. Important for this talk is to know there is a virion, which is commonly referred to as the virus, but it's only the transport form that contains the RNA. Then there are 4 structural proteins, large molecules that make up the virus shell and the virus. Then inside is the RNA that gets read out and translated into a polypeptide, actually several. And these proteins then get like clipped into functional enzymes, and those enzymes are taking over the host cell, numb the immune system, and care for that more virus is made in this foam in the Golgi ER apparatus that is building up inside the host cell. And that's all facilitated from 16 non-structural proteins that are all in one long polypeptide chain and have to be like cut in order to take over the host cell. And then the RNA that has been copied is exported from this foam inside the cytosol, so inside the host cell, and gets packed into new virions. And then for each one that infected of infection, or if you so will, the life cycle. So in total, there are 8 proteins where the function is unknown. In total, there are 28, depending a little bit how you count, but 28 proteins. That is all that coronavirus needs in order to take over the host cell, together with the RNA. Of course, if we can interrupt any of these like steps, for example, cutting the polypeptide chain into functional bits that can take over the host cell, which is done by this protein called main protease. If we can inhibit that and hinder it from cleaving the polypeptide chain, we can interrupt the cycle of infection. And if we interrupt the cycle of infection, a COVID-19 patient may not get more ill. So it's a drug. So an inhibitor to this enzyme is a drug. Most drugs are actually inhibitors to proteins. So here it is with a drug candidate fused to the protein. Another important thing is the spike. I'm going to talk about this a little bit more in detail later, but the spike is what sits on the surface of the virus and allows it to bind, for example, to lung cells. So where do these structures come from? The overwhelming majority, 1,300, comes from RNA polymerase. Comes from crystallography. That are all structures except RNA polymerase. Small number comes from nuclear magnetic resonance, and the rest comes from the newcomer in the field, cryo-electron microscopy, for most part, single particle cryo-electron microscopy. So, as I said, errors in structures are pretty normal. And now we add to this like already complex problem, the pressures of an ongoing pandemic, a lockdown in which the researcher has no good contact to his supervisor or his colleagues. And basically the idea that scientists now need to save the world and quickly find drug and vaccine targets in order to cure the pandemic. That was the situation back in March 2020. And actually for the most part of the last 2 years. So we did what we felt we must. We came together as a group of, in total, 26 individuals to fight the pandemic. Most of us are methods developers in crystallography. Some of them rather big names like Jane Richardson or Kai Diedrichs or Dale Tronrud. Some of us students as young as 23. We came together from spontaneous willingness. There was no like research project that said, right, Andrea, no, you're now gonna lead this international task force and look at all those 2,000 structures that we know. It wasn't like that. It was merely we were seeing this pandemic and we thought it might be fun to improve all the then known 150 structures from the SARS virus. And we thought we'll do this for 3 weeks. And after 3 weeks, our lives would continue normally, and we'd possibly write a little review on how structures can be improved. That was the plan. And actually on this phone, I made a chat group. And that chat group was named Coronavirus Structural Task Force as a joke, right? So this is how these things start. Of course, the thing quickly escalated, and I'm going to talk about this in the end a little bit. So we are methods developers. That means we're specialists in solving structures. We are also specialists in finding and recognizing errors for what they are and to exactly say what can be improved. So we're more focusing on what can be improved than on the error part. And when we find something in a structure where we think that structure really needs to be like adjusted, we write to the original author, tell him, here's your new PDB file, no strings attached. You can just use it. You don't need to cite us, just improve the structure on the PDB so that the fight against the pandemic can continue. We evaluated all published structures. We still do this from SARS-CoV and SARS-CoV-2 and some human interaction partners. We reprocessed and remodeled those structures. And then we did scientific dissemination. So in the last year, we did more scientific dissemination and less structure correcting. And we also do public outreach. So in the beginning, on Wednesday, in the beginning of the PDB week, we get the new PDB entries. We find out which ones are from SARS and SARS-CoV-2 by aligning the sequences. It's not trivial, by the way, because people don't give the right source organism. And then we populate a metadatabase with it, calculate RMSDs, look if there are raw data, which unfortunately there are not always. And then we do an automatic pipeline that runs a lot of like partly customized versions of these softwares that are all validating structures automatically. Unfortunately, though, these tools are great, but unfortunately, you still need an expert crystallographer to look at the structure, an expert cryo-EM person to tell whether something is really wrong. Then here's some manual things. So for selected structures, those that are really of crucial importance to the fight against the pandemic, for example, the structure of RNA polymerase bound to remdesivir, which I later show was what's your structure, we look at them atom by atom. That takes quite a while, and it's also one of the reasons why the task force is so large. Then we disseminate directly on GitHub. So typically by Wednesday evening, the things are up online. We tweet about what we do, 3D BioNotes and MolSSI directly link into our database on GitHub and display our results in 3D BioNotes as well as on the MolSSI, which is a bioinformatics institute homepage. We contact the authors and we make entries to Proteopedia and also on our homepage. And last year, we started writing reviews about the structures that are known and what we know functionally about each one of the structurally solved 18 to 19 proteins that we now know. So here are some global statistics for the corona structures. This is purple is molecular crystallography and yellow is cryo-EM. You can see they're fairly normal resolutions. Our free values are also pretty typical. As I said, the PDB average is about 24. So to our great surprise, the more statistics we do, we find that while coronavirus structures contained a lot of like things that could be better, that is also true for the rest of the PDB, and structural biologists weren't as bad as we initially thought in solving these structures, even though they were at home with their children in lockdown and had really the worst like circumstances and a lot of time pressure. We also give for each sequence statistics like this. This is an RMSD plot where we can see whether, so these are all structures of endoRNAs, and we can see how similar they are to each other. That allows us to say automatically, is this a new fold that we haven't observed previously for the structure? And then on the database, you can find more information on each structure, but I would recommend using the homepage for better access. So there are things like this, and then you can find also blog posts, scientific and public ones. So here are mostly scientific ones, and they are basically the pre-versions of our reviews, which will now come out in a few months. But we also talk about things like how does structure-based drug design work? Why do masks protect against aerosols? How does mRNA vaccine work? So we also talk about things that are more widely related to biochemistry. And if you are doing coronavirus research or just a very well-informed scientist and you're interested in writing a guest blog post, please contact me. We're always looking for those. You can find a homepage at insidecorona.net. And it's not looking like this anymore because we have a new layout since last week. And yeah, this is what it looks like. So you find information about our database, about different quality indicators that helps mostly drug developers to pick out which structure to use, and information about how to evaluate if a structure fits your purpose. And we're soon gonna supplement this with entries for each molecule, which structure we think is like the best one for this and that purpose, like for molecular dynamics or drug design. And yeah, there's also the blog post, but I already showed that. So modeling problems we found. This is an atrocious Ramachanan plot. For those who don't know, all the spots should be blue except for about 2%. So this is a really weird fold. We also had occupancy problems where people have modeled. So green density means that the experimental data are not agreeing with the model and there should be more atoms. This one actually has a model inside, but that model is not at full occupancy. I'm sorry if this is like beyond what some of you understand. It's very technical detail. So if you're not like a structural biologist who has an idea about occupancy and that's now the perfect moment to get yourself a coffee. We also have things like this where you have cysteines looking very weird. So it's very uncommon for sulfurs to just stick out while two other sulfurs are bound. In reality, that's of course a zinc finger domain and there's a zinc missing. So metal centers are really a problem. People don't get them right. We're possibly not doing very well at telling them in software. And we need to improve what we are doing there. We also found metal centers that looked like this. So I don't know if you know this, but zinc usually has 4 coordination partners in water. And here there are 5. And it's of course because the histidine needs to be rotated out of the way, which also improves the bond length. Trans-cis problems. So here's transproline that should be different. And you can clearly see that the electron density indicates it should be different. So we turn these around. There are many cases like this, and we don't know why they happen, where you have, this is a glycoside sugar attached to the protein molecule. And because the chirality is wrong down here, the whole thing doesn't really sit well. If you turn the chirality around, which is not quite trivial to do in COOT, it sits much better. And that like chirality of glycosidic bond wrong error happens a lot. And we still don't know why. We talked to John Aguirre and Paul Emsley, who are like foremost authors of the problems related, and they didn't know either. Here's remdesivir. It's still like a recommended drug, at least in Germany, and I think in the entire European Union against coronavirus. It's an adenosine analog. I know it doesn't look quite like it, but it gets cleaved. So it looks like adenine to the host cell and in particular to the RNA polymerase. So the viral RNA polymerase is a protein whose job it is like a photocopy machine to make more RNA. It has a template strand from the virus and it makes more viral RNA by copying it. So basically remdesivir, which looks like adenosine, is bound by the RNA polymerase to the nascent chain, to the new chain that it's just about making. It gets attached and then it gets ratcheted up into the polymerase machinery, and there it's thought to stop the mechanism. So then the polymerase is basically plugged and it can't move on. And therefore the RNA polymerase is inhibited. It can no longer copy any more RNA. That works very well in Ebola. Unfortunately, the coronavirus has one of the largest genomes known for viruses, and that includes a proofreading machinery, which is able to go back, like it like fiddles, ratchets the whole mechanism back, cuts out the remdesivir, puts in the correct adenosine, and then continues, which is why remdesivir isn't so great after all. But there was this structure which supposedly proved that RNA polymerase binds remdesivir, and we can use artificial intelligence. So I'm also developing the program HARUSPEX, which is CCPM, to identify secondary structure in cryo-electron maps. Yeah, so here you can see the map, and then the network can color it automatically according to secondary structure before a model is built. And then you can build in the model and see there it fits, right? So yellow is RNA, and you can quite see how that fits nicely. So if you're interested in using that, let me know. It's in CCPM distributed. But here is that map. So that's the reconstruction map of RNA polymerase. The template strand is green. The nascent chain strand is orange, and remdesivir is covalently attached down here, but Pymol didn't show a bond. In the active site are also free magnesium ions and the diphosphate, which is of utter relevance to people who want to develop drugs like remdesivir. And the original authors had modeled them in that's really in the middle where new nucleotides are attached. And when you look at the density, something becomes pretty obvious. And that is that we are not really believing in, or what does mean believe? Yeah, there is not really much evidence for the existence of these diphosphate because they're not covered by density. Only that magnesium has a little bit of density. So when we improve the structure, we remove those. And that is what we wrote back to the authors, and they then amended their entry in the PDB. And that error actually cost a lot of money, but it would have cost a lot more money if we wouldn't have removed it. So in the end, possibly there will be no paper saying the work of the task force really helped develop this drug, or perhaps there will because people are nice. But it's an important thing to do. In the end of that same structure, and 17 like it, there was also a C-terminal out of register shift of 9 amino acids. So all RNA polymerase structures from SARS and SARS-CoV-2 had this 9 residue out of register shift. So here's correction in Isolde. So Tristan Kroll, who develops the software Isolde, which allows you to work on your structure while it's wiggling according to molecular dynamics. Here, so he marks it up and he releases it, and it flops into the correct position very nicely. So I'm sorry, I could watch this for very long. We are twitching these things also that people can see us working on structures from time to time. But this out of register error never occurred again in any structure after we had made it public. And of the 17 structures, 12 were amended. If you want to know more about this case in particular and others like it, we published a paper making the invisible enemy visible, which is also the main citation for the task force work. And we had a lot of partners and downstream users over the time. So by April 2020, we thought we just do this and it'll be fun. And by June 2020, this has escalated quite a bit because the European Union decided to write out 60 million euros on the basis of our structures. The media wanted to talk to us, and all these collaboration partners were suddenly there. And the task force grew and grew. In the beginning, we were, of course, only like 5 people. After week 1, we were perhaps 6, but then we exploded. So it was a very exciting time, and individual labs as well contacted us. IBM Open Pandemics, Folding at Home, used 2.4 exaflops of computing power on our structures to do mostly molecular dynamics. So that was and still is really cool for us, that the fact that the structures that we improve and the knowledge we make and consolidate is really helpful for other scientists out there and enables them to fight against the virus, but also to better understand it. Because that's also something that now after 2 years becomes more and more clear. It will not be done when we find a good drug against SARS-CoV-2. We actually need to be prepared for the next pandemic as well. So what does the virus look like is an interesting question. And you all may know this model from the CDC. but that has a bit of a problem, or actually several. So this is the model that we published in 2020. And if you compare the two, oops, you can see the spike heads actually have stalks, and the surface is a little bit less coarse, and there are many other proteins in the surface. So we published this and made it available so people can use it. We also showed this inside view. I'm going to talk about this in a few minutes in more detail, but I quickly wanted to show you the model. So this is the model from 2020 here. I hope you can see my picture. Can you see my picture? Perhaps. Chat?
Yes,
oh,
we can.
Yeah, good. So we all even made like little antibodies, so you can like show how the antibody binds. We also made a bigger spike model. And if you want your own, you can download them from Thingiverse. And we also have instructions on the homepage how to print these things with a 3D printer. And then so this is basically very good for school demonstrations where you show like, and then your body needs to make those antibodies. So they recognize spike, but it doesn't matter what the spike is attached to. And that's how vaccines work. And we have a very new one. This is the new model. So it's a little bit smaller than the old one. And it's based on electron tomographic data. So we know the spikes are quite flexible. Meanwhile, the diameter of the virus is a little bit smaller, and there are only about 20 spikes. Numbers vary from 8 to like 30. This is a rhinovirus for size comparison. So you can see coronavirus is really huge. It's got one of the largest RNA genomes in positive strand RNA viruses. And this is already available on Thingiverse, but we haven't done the press release yet. But there will be a blog post, I think, next week. So I'm quickly gonna stomp with you through the life cycle. So we've done some biology, and I can talk about what we don't know, because one of the lessons we have from this is all the stuff that we don't know yet about the coronavirus. So here's the infection cycle of the virus. And first there is the spike protein, which allows the virus to enter the host cell. And we made an animation of that. And what I can tell you is when you make such an animation, you understand how much you've not understood. So here's the virus with the structural proteins, membrane, envelope, spike, and nucleocapsid. And inside is the RNA genome. And that now binds to a host cell. So the host cell is shown here on the bottom. It's that big thing. And it has ACE2 receptors, which are expressed mostly on lung cells, which is why the virus binds to lung cells. And here comes the virus. So we suppose it's binding with 2 spikes. It could be 3 or 1. We don't know exactly, but it recognizes this ACE2 receptor. And that's sure, we know that. And then it gets cleaved by surface proteases such as Tempress2, for which, by the way, we have no structure. So you can see they have these like long floppy bits, and that's because we don't know what their structures exactly look like. But they cleave the spike protein, and then somehow magically, the spike protein ends up being tethered to the host cell. And that bit we really don't understand very well. We think it must be refolding. Wiesler basically formulated this hypothesis, I think, and then it's energetically unstable and refolds in a way that it drags the virus onto the host cell, and the 2 cell membranes, so the virus membrane and the host cell membrane merge, and the RNA can then be interjected into the host cell and start the infection. So this animation was made in collaboration with Janet Iwasa from the University of Utah, and is also free as under a Creative Commons license. So if you want to use it, be our guest. So we don't know exactly how entry works, but these are the bits that we know. From here, the RNA is translated by the ribosome into the polypeptide chain. And before that, it's pegged by the nucleocapsid. But how exactly that works, we don't know. So nucleocapsid has 5 domains. an RNA binding domain and a dimerization domain, which are structurally known, and then lots of bits that we don't know and which we think are intrinsically disordered. So they are just not forming a stable fold, at least not in the absence of RNA. And there has been this early hypothesis how the nucleocapsid here shown in gray is packing the RNA. However, tomographic results already in 2020 showed what's called the eggs in a basket hypothesis, where nucleocapsid attaches to RNA in a way that it forms little balls. It could be that reality is even more complicated than that. So how does nucleocapsid exactly bind the RNA? We know all 5 domains are interacting with RNA, but how? Is it folded completely under certain conditions, and how is the RNA packed? So the RNA is 30,000 base pairs. It's really long. And that would mean in the terms of this model, that would be 10 meters and 1 millimeter in diameter. So how does the virus pack this? And what we also know is there's a pseudoknot, which is a fold in the RNA. So the RNA can fold as well, albeit not quite as stable as we'd like to. So the ribosome, which translates this, arrives at the pseudoknot, and most of the ribosomes fall off. So they have only produced the open reading frame 1A. But 1/4 of them in vitro stays attached, goes back a bit, like -1 base pairs, and then with a different reading frame continues to make the rest. So the virus has somehow managed to do like this 4 times as many as the entire length polypeptide, which certainly helps regulation. But it would be kind of hard to believe that in 30,000 base pairs, that's the only functional bit of RNA. I suspect the RNA has a lot of functions that we cannot analyze and we don't understand, and it would be totally good to understand them because they're crucial. This has been shown in studies. So when we interrupt the RNA packing, like with small interfering RNAs, like little bits that attach and like make the fold broken, the virus can't replicate. So they're an interesting drug candidate that's not talked about much. We know it's a not free pack. It's got a distinct fold that is very conserved. It's about 10 times as conserved as the RNA fold in Zika or Ebola or HIV. It forms long range duplexes, higher order junctions. So here's a picture of like 2D fold. It's mind-bogglingly complicated, and we really, really lack the proper machinery and methods to analyze this in 3D. And I would love that because it would tell us so much. And we know the duplexes persist in the host, so they have regulatory function. It's very stable, evolutionarily ordered. It's got evolved to be like that. And it's globular because the virus is globular, the RNA needs to go in. So as a consequence, it's globular. So what are the RNA functions and how is ribosome recruited onto the RNA? And by the way, if you expect me to answer these questions in this talk, I'm very sorry. These are open research questions. These are lessons we learned. We basically came out of coronavirus research with more questions. But I'm hoping that someone is in the audience who may find this interesting. So here's, by the way, the main protease, which then splits the polypeptide chain that has been made into functional bits. So far, the only inhibitor that is a drug on the market is from SARS, but there have been like lots of studies about main protease. There are 450 or more known structures of main protease bound to drug candidates in the PDB. So that was a huge effort. How far off are we a drug? I would like to know. And here is NSP3, which is a very long protein with 2,000 residues, many functions. The students call it the Swiss Army knife. It's got 17 domains, 2,000 residues, and we really don't know exactly what it does, except it also cleaves the main polypeptide chain. And we would like to know not only its functions, but also the overall fold of this like hulking monster of 2,000 residues. And why is it making sense for the virus to do such a large thing? So then happens export, and we're not quite sure how packing works either. So we know NSP3 is really important, NSP4 and the E protein are all essential to packing functional virions, but how it works, no one knows. And then it gets exported. And here we are, life cycle of SARS-CoV-2, and some of the questions we still can't answer after 2 years. We also don't have structures of these proteins, which except TMPRSS2 are all viral proteins. So if there are any structural biologists out there, please be my guest. We'd like to know. In the end, I'm going to talk for like 5 minutes about what all of this meant for us. So you have to imagine we were a group of perhaps 20 younger, mostly younger researchers, postdocs, PhD students, junior group leaders. I was a junior group leader for less than a year when this all started. And we came together and we got funded by the German government a little bit, but our funding is running out now in June with no like renewal in sight. So here we are. And for most of the pandemic, of course, we were not all in one place. Up until today, my group is scattered across Germany. So even the people I employ are not all in Hamburg because we managed to work efficiently together from home office, which is not our favorite mode of working, definitely not. But we have Zoom meetings. For most part, we had daily Zoom meetings. Now we are at 3 meetings per week in the end phase of the project. But we kept in touch both socially and over instant messengers, as well over Zoom. We had large press and outreach things. So television teams came to talk of us. And you may think that's fun, and I can tell you it definitely is. But the kind of pressure that you can ruin your entire reputation in one interview is weighing very heavily on you once the camera is going. So I have also been, so I've been explaining to the public what we are doing, what the virus looks like, how structures will help to develop drugs. My students also took part in a lot of this, which I thought is fantastic. So I always try to bring them in the front of the cameras and make them put videos on YouTube and like talk to the public, because I want our research to be tangible. The general population needs to touch what we are doing, and that will help them to get a sense of control over the pandemic. So that's of course like a high goal, but you know, if anyone, it must be us. We are the researchers, we're the people who really know, so we need to talk to them. And you can see like how a biologist fights from home office against Corona. And this is me on German breakfast TV, which was a really interesting experience. Turns out very many people watch German breakfast TV. We also did a lot of social media. So Lisa, who later became our web designer, sent us this picture she made of the entire group just from the photos on our homepage. And of course, there were lots of group chats, and we also had things to cheer us up in between. I mean, please don't get me wrong. Many of us had COVID, and some of us have lost people due to COVID during the last 2 years, even in the task force. And not being able to see your grandma in another country for a year before she dies is horrible. We've all experienced losses, and this pandemic is not something to be joked about. But you cannot sit in home office under so much pressure with all the media and all the research money that's spent on our result without some release. So I was regularly asked to print stickers as a reward for things people did. So we did that. Some people sent us masks, which say crisis hero. We have a cactus, which is called Corinna. We went on YouTube and Twitch and Twitter. And we also used virtual spaces. In this case, Galver, we are now changing to Work Adventure. And we play games in the evening occasionally together. So once per month, we are meeting for some social activity online. Here you can see us play Draw Battle. And that means that after 2 years, we forged friendships and community across 9 time zones. So not only do we hope we contributed to the fight against this pandemic and did some work that in the great picture of things may be relevant, we also got to know each other really well and went through this together and became a team. And if you ask me what's gonna be relevant for myself in 20 years, I wouldn't know which one is more important. I am really excited about having met these people and that they follow my lead. It is absolutely huge. So in order to understand the virus and its life cycle, we need to understand molecular biology. That goes for the virus, but I think we also need to improve how we model and calculate these structures and improve structural biology as a whole, so we are more ready the next time. Understanding, however, this virus and its life cycle will help us now with the design of therapeutics, so it's also a short-term goal. We evaluated the structures from the virus with a bespoke pipeline and expert knowledge. We provide context, and very recently, we also started asking the questions that structural biologists haven't yet asked, because we have this unique expertise of we know about all the proteins and all the structural biology in this team really in depth. And here's again the paper. And here are some more challenging questions. So what can we learn from our findings in general? So the errors, for example, we found the thing with the glycosidic bond metal centers. Should we change our attitude towards errors in general? Should we make our software so that these errors don't happen so often? How are we dealing with errors? Should this project serve as a model for other projects? We were bottom up. There was no gray-haired professor involved that said, here you are, 1 million research money. Now go ahead and cure Corona. That didn't happen. So perhaps there are a few lessons to be learned, although I'm not super enthusiastic, because worldwide pandemics don't happen that often, and you really need that willingness to contribute. So the project needs to be most important to the hearts of the people. Can open science compete? We published everything online, and we didn't get as many citations when we published finally a paper as we would have liked. So as long as open science is like that, when you publish something in the internet, people just take it for granted. I think open science can't compete. We'll need a different system to evaluate contributions if we want that. How senior do you really need to be? So the review I'm going to submit today so it's gone through referee it's to be accepted. It's like minor changes. The review I'm going to submit today has been written by a 24-year-old about nucleocapsid. 24, is a bachelor student in biochemistry. Olli Kippes, youngest task force member. I am 39. Many of the other junior group leaders are a little bit younger in the task force. How old do you really need to be to devise a research project and conduct it successfully with a team? Because I think our professors are way too old. I think that people should be elevated into permanent position at a much younger age, because that would give us the freedom to pursue during our most active years research goals that are more fundamental and can be more long-term. What will change in science after the pandemic? Will we be the saviors of mankind, or will it turn out in the end that a lab caused this and we caused the pandemic as scientists? How will people view scientists? They're suspecting we may not be up to good, but they're also adoring us. So certainly people are more split about science than they were before. And what will change in science itself after the pandemic? We've seen many interesting things to do with home office. We've also seen very many interesting things in high throughput drug discovery. That will certainly have its impacts. In the end, I would like to thank Thomas Splettstößer, from whom many of these amazing graphics were, also Lisa Schmidt, who's joined our team recently, as a task force member, actually all task force members, our collaboration partners, scientific fairy godmother Arwen Pearson. And we are looking for students for practicals or thesis. So if you're interested in doing your thesis in Germany, please let me know. This is my contact details, and you can also find us on Twitter, YouTube, and on the internet. And with this, I have arrived at the end, and I'm looking forward to read and see which questions you have. And I'm happy to answer all of them.
Dear Andrea, magnifique. Don't know what to say after that. Now you are my lead. Can I do another PhD in your group?
Yes, sure.
Absolutely magnificent. And really, really nice. I hope that everyone feels pushed by your example. It's amazing in all the sense, not only in the how deep you got this project, but also in the way you are doing science.
It was fun. If science would always be like it's been in the last 2 years for us, I'd totally be in for it for the rest of my life. I mean, not that I'm not, but it was super, super nice because we worked on something that holds relevance. Yeah, it's like we're doing something that's important for humankind, brings the best out in everyone involved. People suddenly are working together, and they're also nicer to each other, which I feel is sometimes lacking in science a little bit. You know, that you're happy and you see the contributions that the other make more than rejecting them. I feel that as a junior researcher, my life is full of like rejections from grants, rejections from position, reviewer comments. We are not praising each other enough. But here in this research, Jose, was amazing.
I also, Andrea, I fully agree with you. We need younger people leading.
Yeah.
Because we became all very fast. So I hope nothing we can do about that, but we should. Let me first open the room for the question, and here is I will read it for you, or you can read it as you wish.
No, please read it for me.
Yeah, very nice talk, and also your final thought on age to become group leader, for sure. Now, related with the analysis of error, have you found error related with the technique or, very good, or related with the resolution? Many thanks.
Yes.
I also have this question for you.
We absolutely did find errors that only happened with certain techniques or certain resolutions that are based on how the method works. So for example, Ramachandran errors happened in NMR structures, mostly this really catastrophic Ramachandran plot I've shown as an NMR structure, because NMR doesn't give enough information to refine them properly. We also found, on the other hand, structures, in particular in low resolution cryo-EM, where the Ramachandran plot just looks absolutely perfect. So there have no outliers whatsoever. And that's a sign that they used Ramachandran restraints, and then the Ramachandran plot is useless as a validation criteria. Good. So we also found that. And we also have like error analysis tools, like for example, Fourier shell correlation, that we use only for cryo-EM or only for crystallography. And like things like occupancy problems, for example, only happen under 2.5 angstrom resolution, where people actually play with their occupancy. If the occupancy is 1 across the board, you don't usually get any problems to do with that. So this is why I didn't say this. It's a little bit, we had no student whatsoever in the entire time of the project ever correct structures that we then send to authors, because the expertise you need of the method, how it experimentally works, how the software works, how the chemistry works, all these things, the expertise you need in order to correct structures and with confidence tell another group leader, your structure is not correct here and here and here. That's something that only like people postdoc and upwards can muster. So people like Nick Pierce or Tristan Kroll or Kai Diederichs, they can say, your structure can be improved here. And they're absolutely 100% confident. But learning all this expertise, it can easily take a decade.
Yes.
So I think that it would be prudent for the entire community to recognize this, make the solution of structures more of a team effort, and value the expertise that's there. As a methods developer, I'm always happy when people write me an email, "Hi, Andrea, I can't face this structure. Can you help me?" Yeah, we're here for you. If you are a professor or group leader, you don't have to know everything about twinning. It's our job. We are the experts. Just recognize the problem is no one expects this of anyone. Like, let's work together and get the best possible structures.
Collaborative work is always the best.
The problem is we have still this first author, last author thing, right? And the Nobel Prize, you can only win the Nobel Prize being one person. There is no Nobel Prizes for teams. So as long as science rates everything on individuals, yeah, people talk to me because I'm the leader of the task force. Apart from me, only 2 other task force members, Tristan Kroll and Lucas Antoni, were ever invited to talk about the task force, although there are 26 of us. Yeah, and this is like, I think our community and society see a researcher being one person, genius, sitting alone, making the great inventions, although that's never how science works.
This should change.
Yeah, I think if we can change that, then we can collaboratively have much nicer results and answer bigger questions. I'd like that.
I have another question here from Armando. It was a fantastic and inspiring talk. Thank you. I found very interesting your final general remarks. Coming back to the particular, what do you think is the most important outcome from the Corona Task Force?
Oh, that's super difficult. I think there are 3 things going from the objective to the philosophic. The first thing that is an outcome is we have now a database of 2,000 evaluated structures that people can use. And the second is within this task force and with the reviews, so there will be a special issue of crystallography reviews where we are going to have 13 articles, and these 13 articles will represent everything we know about macromolecular structures of SARS-CoV-2. So that consolidated knowledge about the virus is the second important outcome. And the most philosophical is this community and the lessons we kind of learned. So it's very fluffy, I know, but it's for me an important outcome that we created this community of younger researchers who are supporting each other and working together and all work in methods development in crystallography and cryo-EM and have exchange. And that will hopefully, if we all make it to professor, which is not really clear because some of us are, actually everyone more or less is struggling to become a professor. So if we end up being all professors, we'll possibly sit, at IUCR 2040 in the back row and go like, do you remember when we were young and did this task force thing? And that community and friendship is, for me personally, also an important outcome.
Another question from AC, don't know the name. Thanks, Andrea, for your talk. Apart from the deposit PDB data, did you get access to the raw experimental data for improving the data processing? I guess so, huh?
We did get only raw data if they were publicly deposited. We could never convince a single author to send us raw data when we asked. We asked several times. There were a lot of data sets, 6W9C possibly being the most prominent case where we would have, but for 6W9C we got the data, but there were cases where we really wanted the raw data, the images for cryo-EM or the crystallographic data. And it would have been so useful because we have Kai Diederichs on our team. We can just reintegrate the things. And we're also well connected with the people in Dials and global phasing, so with Clemens and so on. We know all of them and we can always talk to them and go like, can we improve this? And it would have been great to get raw data for all of these 2,000 structures. So please, if you're a structural biologist, deposit your raw data on Zenodo or SPGrid or proteindifraction.org. Please, because for us as methods developers, this is absolutely crucial, not only to improve your structures, but also to develop our methods. And I'm thinking it's particularly great that synchrotrons are now saying, after 5 years, we're now going to release all the data, because that's finally going to tell us things like how many data sets were actually taken at this beamline and how many PDB entries resulted from that, which is, by the way, as far as we can estimate, only 1%, 2% of measurement time actually results in a PDB entry. That means that the efficiency of our beamlines could possibly be increased. We could measure more structures if we be going about this a little bit more efficiently, but we don't know yet because there are no raw data. Or how many structures are twinned, or how often is the detector distance off, and gives you problems with the wavelengths and the bond lengths. You know, all these questions can only be answered with raw data. So please, let's do like the astronomers and make raw data public.
That should be the next step, absolutely. And 5 years is good, but it's somehow a little bit too long, right?
But we need to give people time to publish their structures.
Yes.
I think if they measure and you would immediately put the raw data online, people could scoop them. But 5 years, if after 5 years there hasn't been a publication.
Yeah, probably it's for nothing. I think yes, we don't have more questions around here. I have many, but because we are already on time, so more than 1 hour, and we don't want to get you more time that we already got. So thank you again, Andrea. It was excellent.
Thanks really for having me.
And the science and the view of the science. I don't know if Pilar would like to say something, or I don't know if she's here.
No.
If not,
well, hello. Really, no. I only want to thank you again because it was a very, very interesting and stimulating conference, and well, it was a very big pleasure to have you with us for this time. Thank you.
Thanks.
Thank you, Andrea. With this, we close the session. So thank you everyone for being here. Thanks. See you in the next one. Ciao.
Ciao.